All right, so before I officially start sharing my screen, I just wanted to say I apologize for the early morning lecture. The thing is that I have a graduation recognition activity in the afternoon that I will be attending. And so for that reason, I had to switch from it being in the afternoon session to it being in the morning session. So I also want to say hi to the members of Dr. Gatto's RAU. Um, it's nice to welcome you to some of our activities. And the hope is that we can be able to bond over different experiences. So I'm in discussion with them to see how we can integrate a few more of our activities, as it will be great to expand the network of the different students and their corresponding projects. All right, so let's go ahead and get started. The point of today's lecture is to give you the theoretical background with regards to cell culturing, because I believe it's a very important part of chemists to get engaged with working with organisms. As much of chemistry is now being explored in the context of living organisms, and focusing on cells as a very simple model, this allows us to really explore different properties that molecules afford, and allows you to learn about techniques that require working in a sterile environment. So I'm going to go ahead and start sharing my screen. All right, so can everyone see my screen?
Yes,
All right. So
yes.
this is a lecture that was compiled by myself and two former graduate students in my lab who are now PhD student, or student, PhD um, winners. So let's go ahead and get started here. So what we're going to focus on is the application of mammalian cell culture for the studies of different things. And what we're going to focus on specifically is the utility of cell culturing. And it's quite useful in the research field. As I already mentioned to you, you learn a number of skills that have to do with handling things at a smaller scale and working with living organisms. But also very importantly, these type of skills are useful in both the clinical and pharmaceutical settings. And so there are a number of types of cells that one could work with. For instance, you can work with primary cells. And what I mean by that is that these are cells that are actually isolated from animals, from living system. And so those cells can be expanded in culture and allow for the study of the cell biology and even about disease, and can be used for production of antibodies, proteins, and vaccines. And so when you're working with primary cells, you have sort of a limited time frame in which you can actually use them. However, there are another types of cell lines that are immortalized. And what that means is that they have been manipulated in order for you to be able to use them in a longer extent of time. So immortalized mammalian cell lines can be grown in vitro for prolonged periods and are commonly used as simple models for complex biology. And so I want to provide you a little bit of the context of what it requires for you to grow cells in a given environment. So one of the really important features is the media in which these cells are cultivated. And so here I'm providing you this representation, an overview of the constituents of media. So the media will typically consist of sugars, amino acids, albumin. Albumin is actually the most abundant protein in our blood, vitamins, minerals, growth factors. So these are ingredients that are key to fortify cells and keep them in a comfortable environment, given that growing on a plate is not necessarily the normal environment in which cells will actually grow. So these media provide them with the essential nutrients for them to be able to grow and replicate. And they try to approximate the content of biological fluids that are essential for the growth, survival, and replication process. So when it comes to the environment itself, so we have the media, but we also have the environment that will keep these cells in a very stable condition. And so what we normally use is an incubator. So here we have the illustration of an incubator. And so the typical parameters that an incubator is set at is 37 degrees with a flow of carbon dioxide to maintain a 5% carbon dioxide atmosphere. And so you may wonder, what is this carbon dioxide for? And so the purpose of the carbon dioxide is to try to maintain a pH balance, so an optimal pH in the cell culture environment. And in addition, the conditions within the incubator allows for a temperature that cells are comfortable growing at, and you supply a certain amount of humidity. So normally in the incubator, although it's not showing this cartoon representation, you have a plate of water, and that is to maintain a certain level of moisture. So this combination of temperature, moisture, and pHs are critical. And so oftentimes you will see that media is supplemented with a dye that indicates the pH in which the media is located in. So here's just the representation of this, where we're using a dye that will tell you what pH range your media is present in. So the typical dye will be phenol red, and it tends to be in this color range. And so what you want to obtain is a pH range that's closer to a neutral pH. And so by the use of the dye, you can see if there's something going on in your system, whether you have an acidic environment, and that could be, for instance, if for some reason the buffering control is resulting in the pH, optimization being lost. You can probably have another factor very similar that's leading to basic conditions. So what one tries to do if you observe that a plate that contains cells has lost the pH control is to try to rescue the cells by quickly substituting the medium with fresh medium and monitoring the color of the substituted solution over time. It's very important to note that because cells are living things, they will be consuming the nutrients. That's an obvious thing. But over time, as they're consuming these nutrients, they're going to need a refreshment of them. So there's only a set amount of time in which they can grow in a given media that you've supplied them. So you do have to replace the media every so often. And the typical rule of thumb is every 3 days. And so it's very important to be on top of the timing when it comes to cell work. So whenever you have to actually work with cells, be very diligent in terms of time. All right. One thing to note is that there are certain conditions, certain cell lines that may not require you to provide a flow of carbon dioxide. So this really depends on the particular cell line that you're working with. And this information is not something that I can readily provide you. It's more that you have to be familiar with the cells in terms of the information provided by the manufacturer. All right, so I want to just quickly leave from this presentation. I want to show you where you might be able to get this information. So let me see if I can exit out of this. And so an example of a site that you can actually obtain cells is the ATCC site. Let's see if I can remove this background here. So ATCC provides a number of cell lines. And so this is the primary source of cells for my laboratory. We tend to work with immortalized cell lines. And so what you can do is, let's say that you know a particular cell line that you want to look up. For instance, we work with the cell line A549, which is a lung cancer cell line. So I will type that into the search, and then I will give a moment to see what pops up. And so here you have some information regarding the particular cells and what's available. So here you even get an idea as to how much cells cost. And so as you can see, working with cells is quite expensive, which is the importance of being diligent about cells and not treating this as a trivial work. All right, so you can click on the cell line. So I'm doing that right now. And then you can see what type of information is provided with regards to the actual cells. You can see with regards to the origin, the tissue of origin, whether or not the particular cell line is cancerous. And then you can see what type of application. So apparently this particular cell line can be used for a number of different studies. It can even be used for 3D cell culturing. So today we're not going to focus on 3D cell culturing, which is a really nice technique to try to approximate the more complex nature in which cells can grow. We're going to be focusing on 2D cell culturing, and I'll explain to you in a moment what exactly that means. But you get some really important information. For instance, where the cells were isolated. So from the lung tissue of a white 58-year-old male with lung cancer. And so you get a lot more information if you scroll down with regards to the conditions in which you should be growing the cells, the type of media that you should be using. So click on all these different tabs to get that information. So many of these websites that are suppliers of cells will provide you the information. Because the key thing is for you to be as diligent and careful about these particular samples, as they are quite costly, and if you're inexperienced, can result in unfortunate use of the cells. All right, so I just want to give you that context of how you can get the information with regards to cells. That's a little bit more precise than just simply looking it up in a published manuscript. All right. All right, so let's talk a little bit about this idea of culturing cells. And so I did mention that one way that you can culture cells is by isolating cells from let's say, from a living organism, from a person. And so the reality is that when we're talking about in the context of cancer, the solid tumor environment is very complex. It's very heterogeneous. And what do I mean by that? I mean that it consists of many different cell types that are composed and form this sort of aggregate. And so here's a representation of this heterogeneous nature of a solid tumor. And so if, let's say, you want to study a particular cancer from a person, because let's say you're interested in doing a personalized treatment, well, you can isolate the portion of the tumor. And what you can do is you can try to extract it to be able to work with the cells directly. So here's just a representation of trying to break apart that solid mass and then trying to cultivate it. And so here's just this idea of a mixture of some of that aggregate and some amount of the cells that have the capacity to be adherent. And what I mean by adherent is that they have the capacity to be able to grow onto surfaces. So oftentimes, it's very important, not oftentimes, I would say it's extremely important to work with cell plates that have the capacity to allow for cells to grow onto them. So these will be special types of plates, and so you have to look at the manufacturing of these plates so that you are aware that they're treated with the capacity for cells to grow onto them. And so let's say that you want to then further separate these type of cells, the adherent cells, from those cells that are primary aggregates that are not going to necessarily stick onto the surface. So then there you can separate them and be able to work with them directly, either in this adherent primary cell form or more in this tumor sphere that you can do what's referred to as 3D cell culturing. So just a little bit of context then, when you work with cells in this nature as adherent cells, individual cells, this would be cell culturing that's at the 2D level. But if you work with them in this sort of aggregate format, then this is what we refer to as 3D cell culturing. All right, so except for certain types of blood cells that can be cultured in suspension, most cells derived from mammalian tissue require adherence to a surface for proper cell division and growth. All right, so here I'm just giving you the representation of a typical cell flask that one may use to grow cells. So this is one example, and for instance, in my laboratory, we do employ these type of flasks, but we also employ a more traditional Petri dish-like flask for growing cells. It really depends on the cell line and what you want to do with the actual cells. So again, very important, if let's say that you're working with adherent cells, again, those are cells that can stick onto the surface, well, then you want to work with plates that have the surface treated to allow for cell attachment. So cell attachment can be facilitated by using synthetic polymers such as poly-D-lysine or PDL, and that allows for the extracellular matrix proteins from the cells, such as collagen, to be able to adhere to these polymers. So they form this interaction that allows them to stick onto the surface, and that's very important. All right, so another really crucial thing is that if you're working with mammalian cells, is to be conscious of the things that can cause for contamination. And so there are a number of different organisms that can unfortunately enter into your plates, but also even worse, into your incubators. And so one of the really important things that if you're doing studies with cells is to make sure that you have a room, an incubator that's dedicated to mammalian cell growth. That's why we're focusing on this specifically for mammalian cells. Now, if you're working with bacteria, let's say that you're doing studies that are for using bacteria for the expression of recombinant proteins, then you need to have a space that is for bacteria. Never work in a given moment with bacteria and mammalian cells because you're likely to contaminate your mammalian cells. So when you're going into a facility that's dedicated for mammalian cells, you need to have fresh pair of gloves, a lab coat that's dedicated to that space. And so I'm mentioning that because I've actually, in washing lab coats, so that each of you has a lab coat that's fresh, that's going to be used in the actual space in which we're doing the workshop. And again, you just want to make sure that that you are entering into a space that is as sterile as possible. So as part of that, you will see that even your shoes are going to be sterilized because there's a floor mat that is going to clean up the surface of your shoes. So it's very important that as much as you can, that when you're working in the space where those mammalian cells are growing, that you try to be as conscious as far as the sterility that you're maintaining. All right, so let's look at these things. So the way these organisms are represented is in terms of size. So you have mycelia that you can actually see with your own eyes. You have eukaryotic cells that can potentially enter into the plate that are not the mammalian cells that you're working with. You have yeast, bacteria, mycoplasma, which is can be a very big source of contamination, and viruses. And so what I'm going to have everyone do so that we can preserve the space that that you will be having your training is I'll have you put on a mask, because that is the space that my students are working in addition to the space of 2 other researchers. So to try to minimize contamination, but also give all of you this opportunity to learn how to work with cells, it's very important that we protect the environment. Okay, so here we get the perspective of the size of these different types of potential contaminants. And we have ways to try to eliminate them if, for instance, one of these organisms do enter into the environment of the mammalian cell culture room. All right, so this requires a proper preparation of the working space. So how do you work with these mammalian cells? So key thing, as I mentioned, is the sterility of the environment. So maintaining and preparing, preparing and maintaining an aseptic environment. So what you will see when you enter into our space is a hood that has been designed for providing a laminar airflow. And so what we do is to open up the space, and we normally keep it UV irradiated so that the UV kills off things that can potentially grow. We turn off the UV, open it up, and we immediately start spraying down with ethanol. So you see that we have this representation of someone wiping down the surface. So what the typical thing to do is to wipe down with 70% ethanol, and to do so all over the surface. And even if you may think that maybe the shield, this glass shield, may have become a little bit contaminated, make sure you wipe that down. Another really important thing, especially because you're working with these delicate cells, is you want to try to avoid putting so many things within the actual hood. So avoid cluttering. Why? Because you don't want to tip over things that's going to splash all over, that's going to get cancerous cells all over the space. So again, try to avoid cluttering as much as possible. All right, so how do we go about maintaining these sterile surfaces? Well, very importantly is to work with tools that are sterilized. One of the key tools will be the pipette tips that you're going to use for transferring solutions. So sometimes we buy the micro pipettes ourselves and they come not sterilized. And so what we do is we use a system called an autoclave, and that autoclave uses temperature to sterilize these materials. And so we use the autoclave in addition to these tips. We use it to sterilize a number of the solutions that we work with, even buffers. All right, but very important to note whether or not a particular solution can survive the temperature at which the autoclave will reach. Now, there may be cases where, if you wish, you can buy pipette tips that already come pre-autoclave and that are labeled as being DNase and RNase free. So that's important. They tend to be more costly. So for cost efficiency, my recommendation is to buy tips that you can sterilize and that you can have a much greater abundance of them. 70% ethanol is key to have a lot of it in the vicinity so that you can quickly spray down surfaces, and you're going to be using it throughout all steps of moving things around in and out of the hood. So even though you may have sprayed the area in general, the outside, the inside, you're going to be using this a lot. Another really important thing is that there are going to be steps where you're going to need to prepare things outside of the hood. And so now you're going to be exposed into an environment that, has humidity and different things like that. So the best way that you're going to be able to pull this off is by using the flame method to transfer solutions, to prepare solutions of compounds, let's say that you want to use to examine in cell-based assays. And so I'm going to show you, we prepared this video to give you a representation of how you can prepare a space and maintain it sterile as best as possible using the flame method. All right, so I'm gonna talk a little bit about the creation of the space, and then I'll show you this video. All right, so very importantly is that you autoclave everything that you're going to work with. And so, as I mentioned to you, we autoclave a number of our solutions, our solvents. For instance, we work with NanoPure water, but we make sure to autoclave the NanoPure water because the sources of water that we have, at least in the Facundo Puesto building, are not great. And so even though we do put them through the NanoPure system, and that NanoPure system has a UV light, still not sufficient to fully sterilize the water. And so we do autoclave, and so we use this really special type of tape that we put on it so that after the autoclave process occurs, the tape will turn black and will tell you that you have successfully autoclaved the bottle and the solvent and solution. Also, very importantly, is the sterilization of the surfaces that you're going to be working with in your lab space. So my recommendation is that when you're working, again, outside of the hood, that you want to wipe down the surface 3 times. You create your space, make sure, again, no cluttering. Make sure that you tell people in your lab that you're dedicating a certain space to do sterile work because maybe they're not aware of it and they may just start putting things in that space that you're trying to sterilize. So very important for open communication. So you want to wipe down all materials that you place in this surface that you have washed with 70% ethanol. You want to make sure that you wipe it down with 70% ethanol, you put it in the space, and then you use a source of a flame, you ignite it, and you make sure that that flame is maintained on in the sterilized surface area, and that you keep everything that you're going to use in close proximity to the flame. So we prepared this video for you last night, so I apologize, there's no actual words, but I will narrate as we go along through it. So we'll click on this. I think I actually already have it. There we go. All right. So as we can see here, I created a space and I'm spraying down some ethanol. I'm taking this big Kim wipe and I'm going to just clean the space here. I'm wiping it down, wiping down, wiping down. And you're gonna see me do this 2 more times. You can see that there's a little label here because that's actually John's space. That's where he'll be working. That's where he probably, if he ever needs to do anything sterile, he'll be able to do it right there in his own little space. So here's my last washing down of the little space there. And now I'm gonna take another Kim wipe. And what I'm gonna do is I'm going to now wipe down the things that I'm gonna bring into that space. I'm bringing a bottle of, who knows what that is, maybe some water, but it's autoclaved. You see that tape with the black, it tells you that it's autoclaved. And I'm just gonna show you this process of trying to transfer a solution from one bottle to another. And everything that I'm bringing in, I'm bringing a pipette, I'm bringing some pipette tips. Again, I'm wiping things down. So you can see that those tips are actually have been sterilized. They have that autoclave tape. Just make sure to add more ethanol just to wipe down. So that thing that you see there, that's going to be my source of flame. And so you see in a moment how I'm going to use it. Here, when I have a big stand like that, what I do, I just spray it down thoroughly. I don't necessarily wipe it down just because it might just take too much time. So I'm just gonna spray it down with some ethanol, and then the container in which I'm going to transfer solution into, I wipe that down. And then very importantly, especially when you're working with a lot of samples, you're gonna bring your marker there to be able to label whatever it is that you're transferring. All right, so now what I'm gonna do is ignite my flame. So we have a few of these in my lab. You see, turning it on. Okay, now that's ignited. So I'm going to try to keep things as close as possible without burning myself. And then I'm opening things up, and everything I'm opening, I'm going to flame it to make sure that I try to keep it as sterile as possible. You can see Carmen in the background working. Okay, so then I flame the container which I'm transferring to. It can be a little hard to manage, but you get the feel for it. I'm gonna try to get that tip as close to the flame as possible without putting it in the flame, because otherwise it would melt. Then I'm gonna take a certain volume, take it careful about it, cover that pretty quickly, transfer it into the Falcon tube. Now I'm gonna close the Falcon tube. And then the very last thing is label. And you want to make sure you're extremely thorough with the whole process. And so we reached this point, and we've successfully transferred that solution using this flame method to keep things sterile. Okay. So any questions so far? I have two screens, so if you have a question, you can always feel free to get on your mic or type up your question in the chat. So feel free to ask any questions along the way. All right, so now let's talk a little about the preparation of the cell growth medium. So many media are actually supplied by commercially. And so it's just a matter of reading the instructions of how you're going to grow up your cells to determine which type of media you're going to actually use. So before starting work, check that the information given with the cell line to identify what media type, additives, and recommendations should be used for the growth of that particular cell line. Most cell lines can be grown in a media called DMEM or also RPMI, but typically media needs to be supplemented. And why it needs to be supplemented is because a number of these ingredients that are crucial for growing cells will not last very long if they're already supplemented into the media. Media is maintained for many, many months. And so one of the things that's supplemented is with serum, right? So serum is basically the fluid part of blood, okay? And so fluid part of blood would not last very long in medium. So that's why you will purchase serum separately, and then you use it, you'll store it in a frozen condition, and when you use it, you thaw it out, and then you'll supplement your media with it. All right, so typically one uses fetal bovine serum, although right now Carmen is going to be working with some breast cancer cell lines and breast cells in general. And it turns out that one of her cell lines requires the use of horse serum. Okay, so bovine cow horse serum, for whatever reason, I was reading up on this, the horse serum is ideal for those cells. If you use bovine, the cells won't actually grow. And so this is very interesting to note. So maybe Isabel, when she works with some cells, she can tell us what type of media she used and what type of supplements. All right, but in this context, typically, you would supplement your media with 10% fetal bovine serum. You would then also add, if the media does not contain it, sometimes media will contain certain addition supplements, you will add 2 millimolar glutamine, and also antibiotics only if required. Sometimes people will do cell culturing without antibiotics because they don't want the influence of the antibiotic in whatever assay they're doing. As you can imagine, antibiotics, small molecules, they can affect certain assays. So you have to read up very carefully if a given assay is not compatible with certain types of antibiotics, so then you would choose not to include them. All right, so you check which culture media and culture supplements, cell lines you're using before this culturing again, and the emphasis being that the media and supplements should be sterile. So what that means is that you have to keep these bottles closed. Do not open them in your lab, in the open space. You need to open them within the hood, or if for some reason you need to do a transfer of solutions outside of the hood, use the flame method. Okay, but very importantly, to keep accepted conditions. So down here we have an example of supplementation. So typical media bottle would be a volume of 500 mils. So to that, you supplement FBS, and so 10% of the 500 mils would be 50 mils, so that's the amount that you would supplement. So you may be wondering, well, like how much FBS itself? Well, you get from the company an FBS bottle, and you literally would just transfer 50 mils from that bottle into the media. So there's no additional dilutions you have to think about. You just literally transfer the bottle provided by the company, you put it into the media. And then you have to add 5 milliliters of glutamine. and then 5 milliliters of your mixture of your antibiotic cocktail. So we typically work with penicillin and streptomycin, but there may be other cell lines that might require a different type of antibiotics, or maybe you're not going to use any antibiotics, so then you would not supplement. All right, so next we're going to talk about this idea of growing cells. And again, these are living things, so they're going to replicate, and especially if you're putting them in a small space, they might start to crowd it. So it's very important that you keep an eye on the cells so that you actually split them. That means that you will reduce this crowding by putting them into another plate to continue growing them. All right, so as part of the process of growing cells is to become very familiar with their morphology. And so it's critical to have a microscope to be able to check on the cells, their morphology, to get a sense of their health. So cells should be checked microscopically every few days to ensure they're healthy and growing. So this is where I say, when you look at the information for the cells, look for a picture of these cells. What should they look like? So if you work with adherent cells, you should have a picture of what they look like when they are adherent to the surface of the actual plate. So this is just a representation of cells adherent. You will see it under the microscope. So adherent cells should mainly be attached to the bottom of the flask. They may appear round and plump or elongated in shape, and they would reflect light around their membrane. Okay, they should also replicate, not the word should, they will replicate at consistent daily rates. So what do I mean by that? Every cell line has a consistent doubling rate. That means a consistent rate at which they will multiply by a factor of 2. And so that's one way that you can tell if your cells are growing well and that they have not mutated, is their doubling rate. So get a sense of that. So as you can imagine, cancer cells are very aggressive. They're going to replicate extremely fast. Non-cancer cells, not aggressive, so they're going to replicate at a slower rate. Get a feel for this rate. The company may provide it, but you also, keep track of the cells and determine if they seem to be growing in a consistent manner. And another really important thing, if you're using media that has a dye, an indicator, then make sure that that color is consistent. Now, a few things to notice and to decide to discard cells. If you work with adherent cells and you see that the majority of the cells are detached, something's wrong, then you have to get rid of those cells, okay? A number of things that could go wrong is, for instance, let's say that you forgot to keep track of your CO2 tank and your CO2 tank ran out and you don't have any CO2 flowing into your incubator, well then, that's going to lead to cell death. If you're working with just cells in general, adherent or not adherent, if they look shriveled under the microscope, grainy, dark in color, get rid of them. Or if they reach this state of no longer growing. So the student in my lab, Lisandra, has had this experience quite often. It's quite since experience where the cells look alive, but they're simply not replicating. So something happened and that stalled them at some point of their cell cycle, and they're not growing, and then they're no longer functional for the purposes you want. All right, so then this idea of subculturing or splitting cells. Okay, so cell splitting, cell passaging, I will often use these terminologies interchangeably. Okay, so the thing is that when the cells are approximately 80% confluent, all right, so here we have new terminology. What does that terminology mean? So confluency has to do with the surface of the cell. So you have a certain space that the cells can cover. 100% confluency means that your cells are covering all the space. So this measure is a relative measure to your eye. If your cells are filling up 80% of the surface, they're adherent, then they would be 80% confluent. And so that is actually an important parameter to be aware of, the confluency, because you definitely don't want your cells to be beyond the 100% confluency, because then you have cells that are not adherent, that are on top of the cells that are forming that primary layer on the bottom. And you're now in a state where the cells simply will not have enough nutrients to survive. But that 80% confluency is that sweet spot, because here we have a depiction of the growth of cells, and it gives us an indication of how the cells are growing in terms of number over time. And that 80% confluency is still within the log phase of the growth of cells, meaning the cells are quite healthy. And that's a nice point for you to split them. So what you would do then is you want to transfer a certain number of these cells into another plate. So you will use a buffer like a phosphate-based saline buffer for PBS. You will remove the media, and the process of removing the media, you just have to be careful so that the cells are not dislodged. I mean, they're adherent onto the surface. Now you'll have them washed with PBS a few times, and then what you're going to do is add an enzyme called trypsin. All right, so trypsin, if I can get my, sorry about that, get my laser back on. So trypsin is an enzyme, a proteolytic enzyme, and that means that it can cleave proteins. And so what it's going to do is going to cleave those proteins that are anchoring onto the surface of the plate. So it cleaves it without harming the cells, very importantly. You use the trypsin for about a time frame of 3 to 5 minutes and use a conservative amount. You put the plate into the incubator, 3 to 5 minutes, to maximize the efficiency of the actual enzyme. And then after that point, you take out the plate, and what you're going to see, you're going to literally be able to see the media now suspended with those cells. Now your eyes can actually see those cells moving in the solution. So now they're going to be in this type of suspension, and what you're going to do is you're going to take that suspended cells and you're going to transfer it into a Falcon tube like this. Okay. Then you have the Falcon tube, and maybe you do a few washes to get rid of that trypsin. You wash it with PBS and centrifugation. You're going to actually see some of this process with Israel, this process of the transferring and washing a bit. All right, and then you're going to have this all the cells now in the Falcon tube, and you're only going to take a portion of those cells to put into a new plate, so that now you have effectively reduced the number of cells from the original mother plate and put them into this daughter plate, and you have a lower number of cells. And so that's very important because now you have these cells at low confluency, but not too low. So typically I recommend that you have confluency at least above 10%. So there's some amount of math you need to do in terms of this process. All right. But very importantly is to be familiar with how long you can use your cells. So working with immortalized cells, either you buy from a company, read up on this information because the company will give you a really important number, which is a passage number. So this process of going from a plate, from the mother plate to the daughter plate, that's considered a passage. And so a number of cell lines have a limited number of passages that they can endure before they start to mutate. You never want to work with cells that are mutated because then many of these results you're going to get are not going to make any sense. So look up that passage number. For some cells, it's very high. From some others, lower than that. And then you basically follow the company guideline. But don't just use the passage number as the law. Keep in mind everything that I've already mentioned to you, looking at the morphology of your cells, looking at whether they were adherent, and get a sense of that. So all those factors that I already told you about, the doubling rate and all those other factors, keep that in mind when you make the decision about whether or not you should discard cells. But the passage number is certainly a very important one. All right, so I'm going to give you an example of a type of splitting that's referred to as 1 to 10. Okay, so just imagine that we are here with the 10 mils of resuspended cells that you took from the mother plate. So what you would do for a 1 to 10 splitting is that of these 10 mils, you're only going to take 1 milliliter and transfer it into a new plate. And then that 1 milliliter, you would use fresh media to dilute this the solution here, and you're going to cover the surface of the plate so that the cells that are suspended in the solution, the media, now have enough media to be able to grow for that period of up to 3 days. So this is referred to as a 1 to 10 dilution because you literally are reducing the density by a factor of 10, so 1 to 10 dilution. Okay, so another really important thing is to be able to count cells. And so here, what we're doing is we have those suspended cells, and we want to count how many cells do we actually have. So what we do is we take a portion of these suspended cells and we mix them with a dye called trypin blue. And we'll do so in this ratio of 1 to 5. Okay, so you're going to have this experience with Israel, this process of being able to count cells. And so what this dye does is very important, is that it actually helps to help you to count living cells and dead cells, because keep in mind that you're going to have a mixture of both, because your cells are going through the life cycle, right? So some cells will be dead in the process. So the trypsin blue, what it's going to do is going to stain those cells that are dead, okay? And that's going to help you distinguish between what's living and what's dead. And what you want to see is that you have more live than dead, because if you have much more dead, it's most likely that the condition in which you did this process to resuspend your cells was not efficient. Maybe we're not careful enough. All right, so we use in my lab a really old school method. At the molecular science building, there's a more fancy tool for counting cells. You're going to use the old school method, where you're going to use this device here called a hemocytometer. And you're literally going to take an aliquot of your trypsin blue suspended cells, and you're going to plate it into these chambers of the hemocytometer. So you're going to see something like this. When you look at the chambers, literally, you have a chamber here and a chamber here. You're going to look at these under the microscope, okay? And it's going to look like this. Literally, it's going to look like this. All right, so you have these quadrants, A, B, C, D, and E, okay? And you see the parameter of how big these quadrants are. Well, it turns out that you're going to focus on 4 quadrants, the ones that are labeled B, C, D, and E. And it turns out that solution that you filled in there, well, each of these quadrants will only contain 10 to the negative 4 milliliters of sample, right? So we're going to keep that number in our head, because it's going to be very important in terms of how we go about counting cells. So this part is going to be a little bit of an active part. So I want volunteers, and if I don't have volunteers, I'm going to call people to help me to answer some questions. All right. So we're going to learn how to count cells, and Isa is going to show you in person how to do this. But I want to give you a little bit of a thinking process before then. So remember that the trypsin blue is going to stain the cells that are dead. So here's just a representation of these dots here that are blue. Those are the representation of dead cells. But the other dots that are yellow, those are cells that obviously they're not stained blue, therefore they are alive. So here, the rules of thumb for counting. Obviously, you want to be consistent. So how do we be consistent? Well, we have to be consistent in which cells we actually choose to count. So you're going to count cells in the quadrant. So this is the quadrant, this whole thing defined by the red. So everything inside of it will be counted except for the blue cells, okay? One thing. Then you're going to count the cells on the bottom boundary line of the quadrant. So you're going to count cells that are here on the bottom line, touching this, okay? And then you're going to count cells on the right-hand boundary line of the quadrant. So we're going to count these cells over here. So what cells do we not count? Well, we're not going to count these that are up here and these that are over here. Okay, so we don't count these. All right. So I want volunteers to help me. Well, let's go ahead. Let's do the count here. I want volunteers to help me to count the number of living cells in quadrant B.
12.
Who's that?
Elizabeth.
Hey, Elizabeth. All right, go ahead.
Oh, sorry. I think there's 12 living cells.
All right, let's see. What do we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, good. So that's quadrant B. All right, so everyone write that down. Quadrant B has 12. All right, so another volunteer. Quadrant C.
13.
13. All right, so how did you do this, Anna?
Counted the ones in the quadrant and on the right hand and bottom side.
Perfect. All right, so we ignored this one and we ignored this one and we ignored this thing here, right? Good, thank you. All right, quadrant D, somebody else.
I think there's 14.
Who was this?
16.
Okay, so that was John and Oscar, I believe. Okay, we got 16. Let's count. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Did I miscount? Oscar, that was you. What did you count?
16.
And how did you count that?
Los puntos amarillos de yellow.
Okay, so does everyone agree with Oscar?
No.
I think it's 14.
It's
14.
14.
Okay, 14. So what did Oscar get wrong? So he counted this one up here and he counted this one right here, right? So it should be 14. Okay, so again, as Anna pointed out, those that are in the quadrant, except the blues, those that are right and those that are at the bottom. Okay. All right, now E, somebody else.
I think it's 13.
13.
Okay, Justin says 13. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay, so we didn't choose these, none of the blues. All right, not these. All right. Okay, so everyone wrote down those numbers, right? Okay, so determining the number of cells in your sample. Well, you want to take an average of the number of live cells in the 4 quadrant. So that's going to be important. That's why I had you write down those numbers. Okay. And then you're going to multiply this number by the dilution factor. So what was the dilution factor? Remember, think back to what I was mentioning to you, that you take an aliquot of your cells and you're going to actually dilute it in your Trippin Blue. And that dilution was a 1:5 dilution. So your dilution factor, therefore, would be a factor of 5. Okay. So you effectively have diluted from the stock, the mother stock, by a factor of 5. So we're going to use a formula down here where the average cell count, and that's the average of the cells in the quadrants, times that dilution factor 5, times there's going to be a correction factor. Okay, what is that correction factor? Well, you multiply by a correction factor to determine the number of cells per milliliter. And recall that each quadrant contains 10 to the -4 milliliters of sample. And what we want to know is how many live cells do we have originally in that mother stock in terms of milliliters. All right, so we're going to take the numbers that we have. So what's the average cell count? Any volunteer?
13.
13. Very good. John, how'd you do that?
I just added up all the different numbers, 12, 13, 14, 13, and then divided by 4.
That's correct. Very good. So you got
Gave it
your
a name.
average cell count. Very nice. Thank you. All right. So then you take your average cell count, you multiply by 5, and then you multiply by 10,000. All right. So what's the answer?
$650. Oh wait, no, 650,000.
650,000. Did anyone else get that?
Yeah, I did.
Okay, perfect. All right. All right. So now we all know how to count.
Me too.
What happened? I said here iPhone.
Oh, sorry. I said me. I also did too.
Oh, sorry about that. Okay, good. Very good. So in theory, you all know how to count, but in practice, these are nicely defined and easy to see. But now in practice, you're going to learn from Isra how to focus the actual chamber, see it very clearly. Okay, so it gets a little bit trickier in person, but still really much straightforward. However, something to note, and I don't have it in my slides, is that let's imagine that this is completely covered in so many cells. You can try to, you all are young, right? Much younger than me. So with your nice, good eyes, you can probably see all these cells very clearly. But in my case, I probably will see too many cells, and what I would decide to do is go all the way back to the mother stock and do a further dilution, so that then I have less cells to count, because with too many cells, there's a likelihood of making a mistake, or even that they may have been clumping that's going to give you an irreproducible number. Okay, so a suggestion is that you want to have sort of a sweet spot of the number of cells, and Isra can give you a nice guidance as to what is a good spread of cells so that it's easy for your eyes to be able to count. Okay. All right, so the very last thing is changing media. So if cells have been growing well for a few days, but are not yet confluent, let's say they have been split 1 to 10. So 1 to 10, depending on where the confluencing was, they're probably a low confluency at that point. Then they will require media changing to replenish nutrients and keep pH correct. All right, so very important that, let's say the cells are growing at the rate that you would expect, but maybe when you did a splitting, you maybe did a little too much, but still the cells are still okay. Then instead of doing another split, what you're going to do is at the 3-day point, you're simply going to change the media so that the cells can continue to grow in a healthy environment. If there are a lot of cells in suspension, when we're talking about adherence or adherent cells, or the media is starting to turn orange because of a change in pH, right? Then media change them as soon as possible. So let's say that even before the 3-day time frame as being the maximum amount of time, you start seeing things changing significantly. Like you're starting to see the beginning of cells becoming loose from the surface of the plate, and the pH is off. Change it immediately, because you have a sort of a certain amount of time to be able to rescue your cells before you have to basically discard them. Okay. To change media, it's very important to try to maintain a consistency of your environment. So remember, the cells are growing in an incubator that's 37 degrees. So you want to warm up your fresh culture media at 37 degrees in a water bath. And so you will see in my lab, well, my students have changed it from it being a water bath to being like this chamber with like these beads. So, you know, students will do as they see most appropriate for their experiment. So you'll put it into this container that will maintain the temperature, and you want to maintain that temperature for at least 30 minutes, okay? That's so that the media, which you will take from the refrigerator, which will be 4 degrees, right? Now becomes warmed up to 37 degrees. You're confident that that is at that temperature. The reason for that is you don't want to shock your cells. You don't want to suddenly introduce them from an environment that's 37 degrees to something that's 4 degrees, because that could cause them to shrivel and die. And you don't want to have something that's a shock reaction that could affect your results, right? So carefully discard the media from the flask into a waste pot containing some disinfectant. And so this is really important, that when you're changing medium, let's say for your plates that contain cells, you might have some cells that are floating, right? And it's good practice to, even if you think these cells are not going to be a biohazard, it's a good practice to make sure that you kill those cells by using bleach, 20% bleach, so some disinfectant, because you don't want this to get on any surfaces. You certainly don't want it to get onto your hands or anything like that. Okay. Immediately replace the media with fresh pre-warmed culture media and return to the incubator with the optimal CO2 and 37 degrees temperature. Okay. So here are some references. I will be providing you these slides so that you have them for your own use. You've also received a handout by email of instructions of the process of culturing these cells and being able to count them. That's the exercise that you're going to be doing in person on Monday with Israel. Before we head out, any further questions, any doubts?
Sorry, what was the cell line that you work with, the lung cancer cell line?
So one of the cell lines that we work with is A549. But we work with a few different ones in my lab.
Thank you.
Okay, other questions?
Yes, can you please explain again how to get the correction factor?
Okay, so the correction factor, that's coming from the amount of sample that's per quadrant. So that's 10 to the -4 milliliters. So we're basically correcting for how much sample you were quantifying in a given space. And we're basically correcting it for the total volume, of sample that you actually put into the actual chamber, so that you can calculate the density of how many cells per milliliter. So that's where this is coming from, this correction factor. Because remember, this is number of cells per milliliter, and each chamber was only looking at 10 to -4 milliliters.
All right, thank you, Professor.
Okay, sure. Other questions?
Sorry, so to elaborate on that, so the number of cells in each chamber multiplied by 10 to the -4 milliliters, that's how you get the correction
Oh, it's
factor?
multiplied by 10,000, Elizabeth,
Oh,
because
yeah.
of the total, the total per chamber is only 10 to -4 milliliters. So it's a very small amount that you're looking at, right? But we're trying to scale this back up to a level of cells per milliliter, right? So we're converting 10 to -4 back to an actual whole number. That's where the correction is coming from.
Oh,
The-
okay, so sorry to reiterate. Okay, so number of cells in the quadrant multiplied by 10 to the fourth.
Exactly. So what is it? 10 to the 4, yeah, right here. To correct for the fact that we were only looking at this small volume, and we want to actually know how much number of cells per milliliter overall. That's correct. Yeah, those quadrants fit a very small amount. So that's why you need a microscope to be able to do this. Otherwise, you would not be able to count this. Let's go all the way back here. You're putting your volume here, and you're looking and you're using the microscope to look at each one of these quadrants. Each one of these quadrants is a very small volume of sample. So we want to scale this to the units of milliliter. Okay, other questions?
I wasn't entirely clear on the concept of 80% confluent.
Okay, yeah, so let me see how I can do this for you. Let's see. Let me try something, John. Okay, well, I'm going to show you something here. Okay, so look, so we have a plate, right? Okay, let's imagine a plate, even though it's kind of ugly. So 100% confluency would be that we have literally covered every space that's available. Okay, so I'm just giving you a drawing of this. Okay, so we literally have all the cells on the space. And as much as I tell some of my students not to do this, sometimes I see them having really confluent cells, cell plates. Okay, so that would be literally covering all that layer of the plate, and that would be, to my eye, 100%. Okay, and so then if we want something that's 80%, well, let's just imagine that sorry, cancel that. We want to have, let's say 80% would be, maybe there's like something like this. We have some of the surfaces not covered by cells. So maybe that to me would be, my eyes, maybe that's like 80%. This is all relative, by the way. Okay, that might be 80%. But let's say that you see something that looks more like this, like so maybe of the space that's being covered is a small amount. That's certainly not going to be 80%. So I don't know, that may be like, let's say 25%. But it's all according to your eye. And it wouldn't look like that. It will look like a little bit more spread over because I'm just erasing these circles that I've drawn. But it's something to train your eyes about imagining the plate being covered with no cells. That would be your 0%, right? With no cells, it would be 0%. And then if you have the whole surface covered with cells, okay, that would be your 100%. And so anything that's between these numbers, you train your eyes to get a sense of what would be, closer to 100% and what is definitely closer to 0%. And so ideally, you want the surface to be in the nice sweet spot of, I would say 70% to 80%, but you definitely want to avoid being completely covered in the surface, because at this point, you may not even can't really see if there's maybe even cells that are like on top of the other cells, and there's actually conditions that are not ideal for your actual cells to be growing. So you want to avoid 100%. You want to be in the 70 to 80% phase, so that you can comfortably passage your cells and know that they're in a healthy condition. Okay, does that answer your question?
Yeah. Also, I was is there like any tips and tricks for like getting to that 80% kind of to sense that 80% confluency, or is it kind of you
Yes,
kind of
so
learn a feel
very-
over time?
Very importantly, John, is this doubling rate. So typically that is provided by a company. It tells you like how often they will duplicate. And so you can get a sense. So let's say that you're at 25% confluency and you have a doubling rate of 1 day, right? That means that by the next day, you should be close to like 50% confluency. It should be that consistent. That's what I mean by being aware of this doubling rate. And I tell that to students all the time because you want to be like sharp about this. If you see that if it's supposedly doubling rate of a day, and then the next day you're still at like 25%, something's wrong. So it needs to be pretty consistent. So not just how they look, but also how they're replicating, the rate at which it's replicating. So use the doubling rate as your guide.
Okay, sounds good.
All right, other questions, other doubts? All right, if we don't have any other questions, we'll have a great day, very productive research day. All right, take care, everyone.
Thank you.
Thank you.
Yeah, you're welcome.
Hey,
Thank you.
hey.
Thank you.